right, so this is as much as I can share with you so far. One more person in here. About the exam. I still have about 5 people yet to take it. A couple of those people I haven't heard from, so they're probably not going to take it. So hopefully by Wednesday, I'll be able to share with you some commonly missed questions. It was kind of like about 1/5 of the class had A's, about 1/5 had B's, but it was just kind of one of those kind of exams. So we have people doing very well. I had somebody get 100%, a few other people get 100% with that slight curve that I applied. So there you have it. You know, over the next few weeks, I know some of you are really needing to get, you know, a certain grade. If you want to work really hard, by all means, come and see me, Zoom with me, and I can work with you a little bit more. The final is 2 weeks from today. So you have kind of a limited amount of time, right, to really throw your all into it. I'm willing to work with you to do that. We're going to talk about the final today. All right, I'm going to go ahead and kind of go in the order that I've posted things. So we're going to finish up our Staphylococcus, move through Streptococcus, and then we're going to talk about the final before we move on. So I want to make sure that I do that today. I'm going to make sure I don't run out of time because that's important information that you need to know how to study and what's going to be on there. All right, so stay tuned for that. All right, let's see. There it is. Okay, so we almost got through Staphylococcus before the exam. So remember our Staphylococcus, we're in chapter 19. This is gram-positive cocci. I'm hoping that the Staph and the Strep, for those of you that are in lab, is a little bit more familiar, but we talked about some virulence factors that make this, especially Staph aureus, is mostly what we've, is all of what we've talked about so far. Even different strains of Staph aureus will have different virulence factors. So that's why we have different manifestations of disease. It depends on kind of the portal of entry that the Staph aureus comes at. And again, what strain that you encounter in terms of what the disease will look like. We talked about a lot of skin infections. To finish up, we are going to mention a few toxigenic infections. Okay, so these scalded skin syndrome is one that I hadn't heard of before, but again, strains that make that exfoliative toxin. So that's a toxin that kind of makes the skin slough off. You know, it does that normally a little bit, but to a much larger extent. So you can see in this patient, you know, the skin's coming off. Remember, that's our first line of defense. When the skin is not intact, we're susceptible to a whole lot of infections. So that can be a tough thing. It's a very painful thing. And when kids get this, they typically fight it off pretty easy. It's one of those infections that as an adult, it can be more serious because that toxin actually sometimes makes the kidneys shut down as well. So it's quite painful and again, more serious in adults. Some strains make the enterotoxin. Remember that entero means intestinal. It's a prefix, it means intestinal. So that can cause food poisoning. This is not a serious infection. It typically runs its course in 24 hours or so. But again, different manifestation. And then another really serious infection, toxic shock syndrome. This, I'm not sure if it was described before the early 1980s, but we suddenly had this huge spike in toxic shock syndrome in 1980. And that coincided with the time a super absorbent tampon came on the market called Rely. And the manufacturers of that tampon advertised that women could keep the tampon in for 8, 9, 10 hours and they wouldn't have leaks. And it did its job, but unfortunately that provides a really ideal breeding ground for Staph aureus. Okay, when you have a nice warm place with lots of blood that changes the normal pH of the vagina. And we saw a huge uptick in women with toxic shock syndrome. What does that infection look like? Very high temperatures, vomiting, diarrhea, a rash that they describe it as it looks almost more like a sunburn. To me, that looks different than a sunburn, but that's the way it's described sometimes. Even mental confusion. Okay, so remember that toxic shock syndrome toxin is a super antigen, right? So causes just way more T cells to be activated than what normally are. So that was what caused this really enormous amount of inflammation and the potential for patients to go into shock. Their blood pressure goes way down, their organs start to fail. All right, so once we saw this association with these super absorbent tampons, they were pulled from the market and we saw the incidence go way down. Today, it's very rare, but we see it in males as well as females. So it's not always associated with tampon use. Right, you should keep tampons in maximum 5 hours. It just can be as a, you know, if somebody has surgery or some kind of wound infection, that's the way you can get toxic shock syndrome today. But again, it's pretty rare. All right, just as kind of to finish up our Staphylococcus, you know, we said that there are something like 40 or 50 different species of Staphylococcus. We spent all that time talking about Staph aureus. Those were all the infections of Staph aureus. So that's the nasty one. The two other most famous Staphylococci are the ones that we talk about in lab. They're pretty mild compared to Staph aureus though. Staph epidermidis is on our skin, mucous membranes, doesn't cause us any problem there. It probably outcompetes other nastier bacteria. It does form maybe more of a slime layer, so kind of a wimpy capsule, biofilms. It doesn't really have any of those virulence factors that make Staph aureus nasty. No toxins, no exoenzymes, things like that. But the one thing that it's known for is that if it gets into your bloodstream, it can cause endocarditis, which is basically attacking your heart valves. So it can cause heart damage, but it's very rare. In Staph saprophyticus, that's also associated with the skin. Also normal microbiota vaginally. So this one is a pretty common cause of urinary tract infections in women of a certain age. Why not when they get older than that? Apparently, if you had a streptococcal urinary tract infection, then you're less likely to get one again. All right, so most women get one when they're in that age group or several, and then are kind of less likely to get it as they age. Okay. All right, so that's our Staphylococcus. All right, moving on to Streptococcus. For most of these slides, I kind of haven't made you write too much just so we can kind of move through things a little bit more quickly. But to begin with here, I'll make you take a few notes. So Streptococcus is kind of similar in appearance to Staphylococcus. It's a gram-positive cocci. And the cell arrangement we describe as chains. So morphology, gram-positive cocci, or cocci, the shape, that's the morphology, again, in chains. So we see this especially when we would stain some cells from a bra. We might see this nice chaining pattern. Okay, like Staphylococcus, they are non-motile. I don't know of any cocci-shaped cell that's motile. Remember what determines motility, if there's a flagella or not. I don't think any cocci have flagella. And they don't form spores. Non-spore forming. And they are generally considered facultative anaerobes. It's their oxygen requirement. All right, so pretty flexible. There are a few of them that are considered true anaerobes, but for the most part, it's actually this is a little bit confusing why we consider them facultative anaerobes, because some of them don't use oxygen at all. They're not bothered by it. They just don't use it. Many of them have only a fermentative metabolism. So if there's oxygen in there, they're just not paying attention to it. So why do we consider them facultative? Because some of them, when they produce their virulence factors, they're more likely to do that in the presence of oxygen. All right, so that term is, maybe you didn't want that deep of an explanation, but that's kind of some nuances of that term. Some of them are considered capnophiles, and that just means they grow best in increased concentration of carbon dioxide. All right, so the brackets refer to concentration. All right, so for that reason, those of you that have incubated Streptococcus in the lab, we want to give them a little more carbon dioxide. We put them in a candle jar. A candle produces some carbon dioxide when it burns. All right, so this is what really sets them apart from the Staphylococci. They're catalase negative. So catalase, remember, is a detoxification enzyme, detoxifies one of those byproducts of aerobic respiration, hydrogen peroxide. So they don't have catalase, but they do have another enzyme called peroxidase that allows them to detoxify hydrogen peroxide with the help of NADH. All right, so you get the oxidized NAD and some water, right? Not as easy to test for. Remember, with the catalase positive organisms, you see the release of oxygen, and that's what we see. Okay. All right, so classifying Streptococcus, by the way, there's about 50 or so Streptococcus, so it's a pretty big genera. There's a couple different kind of ways that we categorize them, and those two ways don't have anything to do with each other, so it can be a little bit confusing. We like to categorize them by hemolysis patterns. So again, this is familiar if you're in lab. When we streak out our Streptococci on blood agar, okay, they have characteristic ways that they look. If they're alpha hemolytic, that means that they have enzymes that will partially lyse red blood cells. So they lyse the red blood cells, but they don't totally, it doesn't, the hemoglobin doesn't totally get digested. So it gets a little bit of a greenish, kind of brownish color change there for partial hemolysis. A complete hemolysis is considered beta hemolysis. The red blood cells totally lyse. You can see right through there. You could read through that plate if it was, you know, if there was something written behind here. And gamma hemolysis, it's a little bit not as easy to see that organism on there, but that just means that the organism doesn't produce any of those enzymes that would lyse red blood cells. So basically the media looks the same when you scrape off a little bit of the colonies there. So 3 different patterns. Every organism is considered one or the other, not one sometimes and one kind of hemolysis another time. It's always kind of one or the other. And the other way that we categorize Streptococcus is based on these Lancefield groupings. All right, so I listed a bunch of them here, not because you need to memorize it, just to kind of give you an idea of the kind of groupings. But these groups, Rebecca Lancefield came up with these. Basically, she discovered in the early 1900s that the Streptococci have these carbohydrates on their surface that act as antigens. So she could make antibodies that would detect certain antigens, and she put them into groups based on, you know, what those sugars were. Okay, so those groups, I think it goes like A to H and then L through S or something like that. Like it's some of the letters are skipped. I'm not really sure why. Some of the groups, there's really one dominant organism that makes up that group. So we're going to spend some time talking about Streptococcus Pyogenes. That's considered a group A strep. So sometimes you see this abbreviation GAS, group A Streptococci. Some of the groups, there's several species that make it up, like group C here. Okay, and in addition, the confusing thing is there's some of our Streptococci that don't have these surface antigens, so they're not included in any Lancefield group. So That makes it maybe not a perfect grouping system, but it's what we do. All right, so this grouping system doesn't have anything to do with hemolysis patterns. They're two separate ways to try to categorize our streps. Okay, and by the way, there are streptococci in animals, and at times strep can be considered zoonotic. So that means that we can get strep infections from animals. We think that's pretty rare, and it depends on what species that you're talking about. I was reading wasn't really a case study, but a little blurb about how they had a family that kept getting strep throat infections, and they traced it back to the family dog that had Strep Pyogenes. And that's where they thought the family was getting strep infections. Again, we think that doesn't happen very often, but occasionally animals will give us strep infections, and we can give strep infections back to animals, again, certain species there. Okay. So let's spend some time talking specifically about Streptococcus Pyogenes. And then we'll mention a couple other streps kind of briefly, but we'll spend most time here. So Strep Pyogenes is beta hemolytic, and it's considered group A. So again, sometimes you see that group A strep designation, sometimes you see group A beta hemolytic Streptococcus. And that abbreviation refers to Strep Pyogenes, which again, it's kind of a little bit too long of an acronym for me. But where do we find this organism? The human throat, the nose, upper respiratory tract, and the skin. Sometimes it's normal microbiota, not very often, not as often as what the Staph aureus would be. How do we pass it? Typically, this is an organism that's passed from one human to another in respiratory secretions. So droplet secretions usually have to be in pretty close contact within a meter of someone to get a Strep Pyogenes infection. And this slide was a little boring, so I threw in the picture from, there's a website called giantmicrobes.com, and you can get little plush stuffed animals of all different kinds of, Ebola, all different kinds of stuffed animals. And this is the Strep Pyogenes one. So it's, a diplococci sometimes, and it's got a spoon and a knife and a fork on there, because as we'll see, that flesh-eating bacteria, that's Strep Pyogenes. That's one of the infections that Strep Pyogenes can cause. All right, so let's talk first about some virulence factors. So things that make Strep Pyogenes successful in establishing an infection and invading into deeper tissues. Structurally, a couple of things. It does make capsules, and specifically hyaluronic acid capsule. Hyaluronic acid is a polysaccharide. We talked about that. Hyaluronic acid is kind of the glue that holds some of our cells together. So this organism's capsule is made up primarily of something that we have in our bodies. So it helps it really kind of camouflage or hide out from our immune cells, because they don't notice it right away as something that's not self. Okay, so that helps it evade phagocytosis. Another structural protein that we talked about back in chapter 14 was M protein. And I showed you the same slide as just an example of an anti-phagocytic factor. The function there, we can go into a little bit more detail now. It inhibits alternative complement activation. So if you remember, that alternative complement pathway was the one where C3 just normally splits on its own. And then I said, if it's in the close vicinity to a pathogen, then it will bind to it, and that will start up the complement cascade. Well, there's a couple other little protein factors that have to be present. And these are proteins in our serum. The M protein actually binds to one of those those serum proteins, and keeps complement from being activated. So by inhibiting that complement activation, it again does another tool to resist phagocytosis from macrophages or neutrophils or dendritic cells. Okay, so and when it's not phagocytized, it's not presented, and adaptive immune systems doesn't kick in as easily then either. There's two kinds of M protein, rheumatogenic and nephritogenic. I bring that up because this is going to play a role in some of the complications that we can see with strep infections. But that rheumatogenic M protein, the antibodies that we make to that cross-react with heart muscle. Okay, so again, we're going to bring that up a few minutes from now when we talk about some complications of strep infections. There can be some autoimmune complications, and the nephritogenic M protein, the antibodies that we make cross-react with the kidneys. And we can get some kidney inflammation, again, as a result of our adaptive immune system targeting those M proteins. All right, a few examples of exoenzymes. So enzymes that the Strep Pyogenes will secrete outside of itself. C5a peptidase is an enzyme that neutralizes C5a. So that's one of the fragments of that complement cascade. Remember, the ones that were A were the ones that caused inflammation. Remember, I said A for allergy, so inflammation, chemotaxis. All right, so if it cuts down on the inflammation, then other cells, aren't going to be called to the area. A streptokinase lyses blood clots and can aid in the spread of infections. All right, so, and incidentally, sometimes heart attack victims can be given some streptokinase that we've made from Strep Pyogenes to break up a clot in the heart that can, again, aid heart attack victims. Okay. And then another exoenzyme, streptodornase, it's a DNase. So if you remember back to one of the ways that neutrophils act on invaders, we talked about degranulation and phagocytosis. Neutrophils can make these nets, neutrophil extracellular traps. So those are basically made of the cell's DNA that's been kind of expunged and used on pathogens to immobilize them. If the pathogen can secrete a DNase to kind of break down or dissolve that net, then it's not going to be inhibited. So one way that the pathogen kind of has a way around our immune system. All right, and then a few exotoxins. Again, it's not every strain of Strep Pyogenes is going to make every one of these. Certain strains will be able to make certain virulence factors and not others. But streptolysins are a kind of hemolysins. So we talked a minute ago about how hemolysins will lyse red blood cells either partially or totally. Well, they don't actually just act on red blood cells. They can actually also act on white blood cells and platelets as well. So what good does that do them? Well, red blood cells are a really rich nutrient source for them, so that can help them, their nutrient acquisition. And when they act on white blood cells, it can actually, if they get phagocytized by a white blood cell, and they're secreting streptolysin, it can cause the lysozymes to release their contents. So before the lysozyme has fused with the phagosome, if the lysosome bursts, then all those digestive enzymes get let loose inside of the cell. And we talked about how some of those digestive enzymes are not going to be active because if the pH is high enough, but others will. So that will cause some damage inside the white blood cell. And rarely, Strep Pyogenes can secrete erythrogenic toxin. So this is another superantigen. So remember, we talked about superantigens are going to activate a wide range of T cells. And the ability to make this toxin is actually carried by a phage. All right, so this kind of pulls back in what we talked about a long time ago, viral replication. If you have a bacteriophage that infects a Strep Pyogenes bacterium, and it inserts, it does lysogeny, so it inserts into the bacteria's chromosome. We know that the ability to make this toxin is only bacteria that have been infected with a bacteriophage, then their phenotype will be changed. Remember, we called that lysogenic conversion, enabling them to make this particular toxin. Okay, so it's fairly rare now. It used to be more prevalent strains that could make this erythrogenic toxin, but that causes a bright red rash all over the body. You'll see it in a minute in a slide, and causes a fever as well. So that's the infection known as scarlet fever. Okay, and again, you'll see a picture of that in just a minute. All right, so let's look at some infections. We'll start with some skin infections, and then add some other ones in as well. Just like Staph aureus, Strep Pyogenes can also cause impetigo, and it's a very similar looking infection. Apparently, somebody that's trained would be able to tell the difference. Sometimes the strep impetigo is a little bit crustier, but again, it's basically the same infection. It stays localized, and you need antibiotics to get rid of that. Otherwise, it will take so long to heal that it will scar. And then a couple of infections that are a little bit more serious, Aerosyphilis, and the next slide, I'm going to talk about cellulitis. So these are two infections that apparently get easily confused a lot. They're both skin infections, but have the potential to be kind of more systemic, so it can invade deeper into tissues. Erysipelas, it looks like raised red lesions. So sometimes it's called an orange peel consistency. So if you think about the peel of an orange, how it's kind of tough, got little kind of pits in it. Sometimes that our skin takes on that appearance. Usually patients have a high fever. How do they contract it? Sometimes after some kind of wound or burn. It can just be even like microscopic breaks in the skin, or you got a patch of eczema that's been scratched a little bit. Those are all just kind of breaking that skin just enough to make a good portal of entry there. And again, lots and lots of complications potentially with that. So it should be treated. Okay. Like I said, cellulitis is a very similar condition. Erysipelas typically is on the face, or I think the legs, kind of same location, hands, face, or legs for cellulitis. And that group A Strep Pyogenes, again, same thing, enters through a break in the skin. Localized inflammation and fever. So you can see this left hand is the one with the cellulitis infection. You can see the redness, the swelling, that looks like it would hurt. Okay, it has the potential to spread to the bloodstream and become life-threatening. So this is something that shouldn't be ignored. Doesn't have anything to do with cellulite layer of fat. I'm not sure where that name cellulitis came from, but again, should be treated with some antibiotics. And the worst of the worst, and I wouldn't maybe necessarily, maybe it's a little bit of a misnomer to consider this a skin infection, but it's going to start out as a skin infection. So necrotizing fasciitis. So the fascia is the connective tissue that kind of holds all your organs together below your skin all the way to your bone. So certain strains of Strep Pyogenes can make exoenzymes not ones that we talked about in our virulence factors, but things like lipases and proteases, things that will break down lipids, break down proteins, that basically allow the organism to digest that connective tissue, that fascia. And it's a very aggressive infection, so it grows really quickly. We can treat it if it's caught early on. The trouble is those early on symptoms are things like, oh, a little redness, maybe a little pain, maybe flu-like symptoms. It's not anything to tell you that you have a life-threatening infection. And then basically, once it's discovered, the only treatment is to kind of stay ahead of it and cut out the infected tissue. Again, because it's so aggressive. So mortality here is about 50-50. All right, so rare, but I have talked to students that have worked in the hospital that have seen this. It's, again, thankfully rare, but it's out there. All right, other Strep Pyogenes infections. Probably the first one that certainly I would think of is Strep Throat. So Streptococcal pharyngitis. Probably everybody's had this at some time or other. Some people tend to be a lot more susceptible to it. Again, transmission through droplets. So you typically have to be in pretty close contact with somebody else that's had it. Streptococcus are not very environmentally stable. So, you know, coughing out on some surface, they don't stay viable very long in the environment. Staph are a little bit better at that than strep are. But it typically makes this pyogenic toxin. So that prefix pyo means pus forming. So one characteristic of strep throat that sometimes is seen, not always, are these pus pockets on the tonsils. I wish that it always made that. And so you could know for sure when you opened up your mouth and looked in your throat if you had strep throat and you needed treatment, or if it was just something viral that you could kind of ride out. Typically will present with a fever, a really enormously painful sore throat. Sometimes you have abdominal pain and nausea, which can kind of make you think it's, you know, just a stomach bug maybe. Often there's swollen lymph nodes. And it can leave the patient opened up to some secondary infections like bronchitis or pneumonia, and if left untreated, as well as scarlet fever. Okay, so for those reasons, if you have strep throat, it needs treatment. And again, not every sore throat is going to be strep throat, but it's not something that you should just ignore or treat with cider vinegar or something like that, because they are some potentially really nasty complications here. So one complication, we could consider this a complication with an untreated strep throat infection, if that strain of Strep Pyogenes has this erythrogenic toxin, then, you know, after a few days of strep throat symptoms, the patient will break out with this bright red rash, right? And sometimes you see this strawberry tongue that's associated with scarlet fever as well. So one of my kids, before she was verbal, had this, and it's pretty rare now. The only, you know, we didn't know her throat was sore until her sister got sick too and could tell us what was going on. And then that rash broke out and we got to the doctor right away. It kind of freaked the doctor out a little bit because, you know, 100 years ago, this was a potentially deadly infection. We think Helen Keller lost her sight and her hearing at 18 months of age because she had scarlet fever. All right, so this was more prevalent for some reason, late 1800s, early 1900s. It's not as prevalent today, probably because we're pretty good at treating Streptococcus infections. You know, we treat them with antibiotics and we get rid of them. But it seems like the strains kind of in circulation now are better at transmission and kind of, again, don't cause scarlet fever. But an untreated scarlet fever then can have the potential of autoimmune implications like rheumatic fever. All right, so this is, don't confuse rheumatic fever with rheumatoid arthritis. That's a chronic condition. Rheumatic fever is an acute condition that can have long-term consequences. Okay, so approximately 3 to 4 weeks after either an untreated strep infection or one that's partially treated. So maybe the patient takes antibiotics for a few days and then stops. It's most commonly in children. What kind of signs or symptoms do you see with this? Sometimes you see these nodules under the skin. And I tried to read up a little bit more about this before class, like what causes these nodules. This is some kind of abnormal growth. I'm not really sure. Apparently this is pretty rare, like only maybe 2% of kids with rheumatic fever will get that kind of around the joints, like the elbow. But they typically have an abnormal electrocardiogram. So it's causing damage to the heart, especially our antibodies that treat that rheumatogenic M protein will cross-react with especially sometimes the joints, but especially the heart valves. Okay, that mitral valve especially can be damaged and leave the patient with long-term heart complications, or maybe have to have a valve replaced or something like that. Okay, so years ago, before we had antibiotics to treat heart valve or to treat strep throat infections, this was more common. If right now we're really good at treating strep infections, our antibiotics work really well, there's very little resistance among the Streptococcus. If that were to ever change, then we would start seeing more of this if we started having trouble treating strep infections. Okay, the other potential autoimmune complication is with the kidneys. So glomerulonephritis, again, that's caused by our antibodies kind of cross-reacting with that M protein and also damaging the kidneys. So the blood flow gets obstructed, increases the blood pressure, and decreases the urine output. And that damage can potentially be permanent. So I've known a few people. One was a student that ended up in the hospital after a strep throat infection. A colleague that I used to teach with, every time he got strep, he landed in the hospital with some kidney problems, all right? So it's rare, but it happens. How do we treat these autoimmune complications? Well, antibiotics aren't going to do a lot of good. They're going to kill the organism, but it's kind of, again, our immune system that we're fighting against. So we have to give immunosuppressive drugs, or immunosuppressive therapy. Okay. And in the case of glomerulonephritis, try to treat the high blood pressure, try to bring that down. There's going to, some edema, some swelling, but there's not a whole lot else we can do other than try to tamp down our immune response to that. Okay, questions, comments? Okay, I just kind of want to briefly question, I kind of cut back a little bit of my strep material here. I want to just briefly mention two other Streptococcus. The ones, again, there's like 40 or 50 different Streptococcus, but kind of the more famous ones, Strep agalactiae. This is another one that you use if you were in lab. It's beta hemolytic and it's considered Lancefield group B. Okay, and it also forms capsules. The capsules of Strep agalactiae are a T-independent antigen. All right, so I'm not gonna let you forget that stuff you learned for the last exam too quickly. Remember, T-independent antigens are B cell receptors can recognize that directly, and the B cells will become activated to make antibody. Okay, it's a fast response. It's not typically as good a one. It doesn't make memory, but also the hallmark of T-independent antigens, young children have a very poor response. to that. They just don't have, they have immature B cells, so they don't get much of a response to T-independent antigens. All right, so, you know, people that are carriers for Strep agalactiae doesn't really cause any problems in adults. It's considered part of normal microbiota, you know, upper respiratory tract, vaginally. The problem here is pregnant women that are carriers of Strep agalactiae. Their babies can pick that up during birth and then can cause some pretty serious neonatal infections like pneumonia, sepsis, that's when the organism is in the bloodstream, or meningitis, very serious. All right, so with good prenatal care though, we can avoid this. Women, I think late in their second trimester, early third trimester, are tested to see if they're carriers for group B strep. And if they are, they're given some antibiotics to try to get rid of it. Okay, so good prenatal care and we can avoid some of these complications. All right, and the last strep that we'll mention here is an alpha hemolytic streptococci, and that's Streptococcus pneumoniae. Okay, so it often presents as a diplococci. We got a nice little transmission electron micrograph there. And I decided there's a lot of different Virions factors that this organism kind of relies on. I decided to kind of skip over the Virulence factors and just talk about disease. This is the organism, if you remember back when we were talking about horizontal gene transfer and that experiment, Griffith was the last name back in the 30s that he did to prove that transformation can take place. He had his rough strains of Strep pneumoniae and his smooth strains of Strep pneumoniae. Like you would guess, the most common disease here that this organism, well, maybe not the most common, a very serious infection is pneumonia. It's probably the most common organism that causes pneumonia in humans. So lots of other ones can. We talked about how Staph aureus can cause pneumonia. There's quite a few other ones, but this is probably the most common one. And doctors can usually diagnose this, you know, just by listening to the patient's breathing. It can cause cyanosis. That means the extremities are starting to turn a little bit of blue because their patient's not getting enough oxygen to extremities. It's very treatable. Typically, the patient will be given at least 2 different antibiotics, you know, maybe a beta-lactam and tetracycline, for example, in combination. So 2 different antibiotics that work different ways to try to make sure they're knocking out this infection. Strep pneumonia can also cause ear infections, particularly common in young children because their ear canal is very narrow and it doesn't, you know, drain as easily. Okay, and often when a child has some kind of viral infection, leaves them a little bit more open to getting a ear infection as well. And it can also, strep pneumonia can also cause meningitis. Any kind of bacterial meningitis, people that survive from that are often left with, you know, debilitating things like retardation, blindness, or deafness. So, of course, that's extremely dangerous as well. All 3 of these infections, though, are less common now due to the pneumococcal vaccine. Okay, that vaccine is marketed as Prevnar 13. Okay, you might have heard advertisements for that on the news, or especially during the news. Lots of old people watch the world news. So they're trying to reach, you know, that 65 or over population that they're trying to get people to vaccinate to try to, you know, they're a population that's susceptible to having very severe pneumonia. This vaccine is also given to babies. So babies less than 2 years old. And again, adults greater than 65 are the ones that are recommended to have this. Sometimes they call it the pneumococcal vaccine. It's given as a series of 4 different shots. They try to get it in before babies are about, the 4th one, before babies are about 15 months old. Okay, and that not only protects them from meningitis, but we're seeing a lot less ear infections in young children that are vaccinated with Prevnar 13. Okay, so I was looking, let me see if I can bring this up again. Yeah, so I was looking a little bit, I was researching this a little bit right before lecture, and I didn't post this, but I found this graph that the CDC has on their website up here that talks about, of course, Prevnar 13 was introduced here in about 2010. So fairly recently, none of you would have had that when you were babies. But back about the time that you were probably very young, Prevnar 7 was introduced. So what are the difference between those PCV 7 vaccinated against 7 different kind of variations of strep pneumonia, and they came out with a little better vaccine in 2010 that would protect against 13 of the more common variations of that organism. So you can see the incidence in pneumococcal disease. So again, it's mostly ear infections, also pneumonia, sinus infections to some extent as well. I didn't mention that before. So they were quite prevalent, you know, at the time that you were young, and maybe some of you got that, and the incidence really fell way down here. Of course, this is cases per 100,000. Okay, I know that when my kids were little, they were just a little bit young to have gotten in on that, but it was very prevalent. You know, any kid that was raised in daycare, you know, they all had ear infections all the time. And so this is a great way to cut down on the use of antibiotics because you're not having to treat kids for ear infections all the time. All right, so just thought that that was interesting. Okay, all right, I think that's all I had for you about Streptococcus. Any comments about Streptococcus? Questions? Okay, before we move on, let's talk a little bit about what the final is going to look like. All right, so it's a week from today. So you've known about this date ever since I published the syllabus in January. So make sure you have work off that day. I was thinking about not offering this online at all. The way things are going, I'm not going to be able to do that. I already have students that are quarantined through finals week. But I really want to cut down. I really want, if possible, you to come and take this in person. And that really will be to your benefit because there's part of the exam that's a little bit easier to take in person. Not impossible to do it online, and we will have to do that. But I know, I would think there would be less, you know, sometimes students have like 3 on one day and they want to reschedule, and I'm always willing to do that, but there may be less of that because there's less exams in person. So if you do want to schedule and make up, just kind of be thinking about that. I'm sure I'll have to be giving some throughout the rest of the week, but as many of you as possible, let's get in here on Monday. And of course, if the university would go online or something like that, all that would change, but let's plan on that now. This final will be composed of 3 different parts. All right, so part 1 is going to be new material. All right, so starting with vaccines, the Staphylococcus, the Streptococcus that we've done so far, through whatever we get through, that's going to be part 1, multiple choice. Okay, so let me talk about that part. So this is what I am anticipating covering. Okay, and I did post this in your PowerPoints if you didn't see it yet, but I'm hoping to get through these bacterial pathogens this week and then do the viral pathogens next week. All right, so we've done staph and we've done strep. I'm going to say already that we're not going to get to Bordetella. All right, whooping cough. I already took those questions out of the homework last Thursday. There are a few of you that had done it already, so I apologize. That's great that you learned a little bit about whooping cough, but I just don't think we're going to get to that this week. All right, so I'm going to say probably almost for sure 99.5% that we're not going to do Bordetella. All right, so Basically on Wednesday, I'm hoping to get through the tuberculosis and the Neisseria. We'll see how far we get through Bacillus today. We're going to get through at least some of it. Okay, and then next week, again, the viral pathogens. So what do I kind of expect you to learn about these different pathogens? You're going to want to, sometimes it seems like I'm just walking through and talking about interesting things about them, but there is some organization there. I want you to know the morphology of the organism. So for the staph and the strep, those are round cells, they're cocci. The gram reaction, gram positive or gram negative. So that's kind of a basic thing to know. For the viruses, you'll want to know if they're DNA or RNA viruses. And, you know, maybe do they have an envelope around them? That seems like a kind of reasonable basic information to know about them. We'll talk about disease manifestations. So I might describe some kind of infection and then say, well, which organism caused this? All right, and that would be, you know, I'll give very specific information there. Probably the thing that's the most not familiar to you when we go through them are the virulence factors. So that might take a little bit more time to go through and kind of understand what those are. So, you know, capsule, specific exoenzymes or toxins that the organism can make. Treatments, in some cases, we'll talk about this more than others. I will usually try to talk about maybe any antibiotics that we use, but I would say don't memorize the antibiotics because my information might not be totally up to date, and that kind of is always changing a little bit. So we'll talk about that a little bit more probably with tuberculosis, what kind of antibiotics are used to treat it. But in general, you know, can we use antibiotics? Does that do any good, or is there something else kind of general there? And then you should know, is there a vaccine? So for the Strep Pyogenes, we already talked about the Prevnar 13. There's, you know, sometimes the name of the vaccine, it's not kind of intuitive what infection that protects from. So that might be something good to know as well. Okay, so that's the new portion of the exam. And then there's a cumulative portion. So this is going to be a little bit less than a third of the exam. And this is also going to be the hardest part, the multiple choice that covers the whole semester. Okay, so I'm still tweaking the final exam in terms of number of questions here. It might be a few less than 30. I have to ask you some cumulative multiple choice. 11 of these questions are questions that we ask everybody that takes this class. So we use them as assessment questions. So some of these questions I've not made up myself. And sometimes when I look at some of them, I think, oh, this is not really the greatest question, but there is a best answer for each of them. And they are things that we have covered. So just keep that in mind if there's one or two that you think, what is this? I don't get this. Like I said, I'm still tweaking these questions and I'm going back through and I'm kind of thinking, well, can I make this question a little bit more general? I try to ask you about things that we talked about several times and not just something that I barely mentioned, you know, 2 months ago. Okay, so to give you a little help with that, in terms of these multiple choice questions, I'll say that I haven't asked you anything from that history on the very first day. So Pasteur, Koch, Snow, any of those guys. And I haven't asked anything concerning the fungal terminology from chapter 12. So we talked about a lot of terms like Castoria and Mycelium and mycorrhizae that we never really talked about again. I did less of that this semester, so that might not be a huge help. Also in chapter 12, that was our eukaryotes. So we talked about protozoa and we didn't mention algae very much. but it's reasonable to understand why we classify something a protozoa? Why don't we classify it a fungi? Why do we call it a fungi? Why is it not called a plant? You know, the difference between bacteria and archaea. So don't forget those just kind of taxonomy things, like what makes a fungi and not a plant, okay? Fungi don't photosynthesize. That's the main thing there, okay? I won't ask you any questions about specific antibiotics. So I know that you just learned those for exam, all those, whatever, 14 or 15 different antibiotics. I won't ask you anything specific about those. I might ask you something about how antibiotics in general work, or what does it mean to be narrow spectrum, or something like that. But I won't ask you individually, or I won't ask anything specific about those antimicrobial chemicals from chapter 9. Again, we didn't really do that many this semester, so that might be less of a break. I cut some of that back, but the difference, you know, is halogens or detergents. Okay, I'm gonna leave that, anything about that off. And when I looked back at some of the questions that I've asked before, I tend to ask a few more about the chapter 5, chapter 7, and 15 and 16. So I know that those seem like maybe the more difficult chapters, but again, think about kind of big things that we can take away. Those are the kind of questions that I like to ask. All right, so I'm not sure if that gives you a lot of help. Sometimes people ask me, how do I study for the multiple choice cumulative part? And I would say, I probably wouldn't spend a whole lot of time studying on that. You might go through, you know, the chapter reviews at the end of these chapters and kind of read through that a little bit. That might be helpful. But I think your time is probably better spent making sure you are really up to date on the most recent stuff, and maybe studying a little bit for the matching. So the third part of the cumulative here is matching of terms. And you can see here, 55 points there. Well, it actually figures out a little bit more than that point-wise because it's out of 150 points. This is the part that students do well on. So sometimes I've had students say, this is overwhelming. I don't know what terms you're going to use. True, but let me show you an example of what the matching will look like. All right, so I've got 4 pages of matching. 3 pages have 15 terms and 16 descriptors. The last page has 12 terms and 12 descriptors. All right, so if you look at this, you know, you really only have to have kind of a basic understanding of what that is, because all these terms, most of these terms don't have anything to do with each other. So, you know, if you don't know the difference between a coccus and humoral immunity, you know, those are really different things. So you should be able to, you know, hopefully kind of narrow it down. In some cases, okay, so I've got cell-mediated immunity and humoral immunity. Okay, what's the difference between those two things? You would maybe be two of these that you might get mixed up, and that would be really understandable. Or, you know, the difference between the TCA cycle and the Embden-Meyerhoff pathway. Remember, that's another name for glycolysis. Okay, I can see how you get that confused, but you should be able to really narrow it down. And if you're doing this in person, you can say, oh, okay, well, I know this is M. Okay, so now I can cross that out, right? If you're doing this online, I found last semester that it was a little more confusing for students because they weren't able to cross things out. I do have a way to do this online, but in some cases, I think students missed more than what maybe they would have if they were doing it in person. So especially for the matching, I think it's good to do this in person. And again, for 3 of the pages, they'll just be 1 extra. So, okay, are there questions about that? Again, and I posted this, you know, practice. There'll be different terms. I did bold type terms in your learning objectives this semester, so that might be one place to start, to look back at anything that's bold typed, or you know, end of chapter review type of things. Okay, it really kind of comes down to if you've been kind of studying and kind of understanding things as you go on, you will remember kind of the basic idea. If you've been memorizing things and forgetting them, that makes this a whole lot harder. Okay, so again, don't be afraid to ask if you have more questions about what that's going to look like, but those are kind of the pointers that come to me right now. Again, I know that's only 2 weeks away. All right, well, let's move on. So how have I decided what pathogens to talk about and which ones to skip? Sometimes I do a little different ones every semester. So we're still in that chapter 19, talking about gram-positive bacilli. I find that a lot of students, especially lab students, we get in a habit of thinking anything that's round, anything that's a cocci is gram-positive, and anything that's a rod is a gram-negative. Because in lab, that's the organisms that we work with. So I like to talk about some organisms that are gram-positive bacilli. Okay, and a lot of you that are in lab this week, that kind of dovetails really nicely because we're going to do a spore stain in lab this week. We're spore staining a gram-positive bacilli. We're not using a bacillus, we're using a Clostridium. I also want to talk about a Clostridium. I don't know if we'll get to that today or not, but okay. So the bacilli, this genus Bacillus, that's one that we commonly use in lab, but we haven't had as much exposure to that this semester because we had to cut out some labs. But when you grow that up on a plate, the colonies of Bacillus are really kind of irregular and they're kind of raised up a little bit. They're really kind of cool looking colonies on a plate, really easily kind of recognizable. In general, this is a genus that's considered environmentals. So where do you find them? Not typically on the body, although some of them are pathogenic, only a few of them. Mostly these are organisms that you find in the soil. Okay, so they're soil microbes. And in fact, some of them are considered saprophytic. Oh, let's see the P. Okay, so if you remember, that's a term that we use to describe fungi. We said most fungi are saprophytic. That means that they, as their nutrition source, they feed on like dead and decaying organic matter. All right, so that's mostly what our bacilli eat. There are a genera of aerobic organisms, so they are catalase positive. And in fact, that's how we tell the difference between Clostridium, a closely related gram-positive rod. The Bacillus are catalase positive, the Clostridium are catalase negative. There are a few of them that secrete antibiotics, like bacitracin comes from a species in that Bacillus genus. And what makes them really famous is that they form endospores. Okay, so here's an example of a spore stain down here at the bottom. Okay, we stain them in a way that the spores take up a green dye. So the little green ovals are the endospores, and the vegetative cells take up a red dye. That just means the metabolically active cell, part of the cell. All right, now it's important to understand that they typically are going to sporulate in the environment, not in the body. There is one exception to that, but for the most part, when that organism is inside our body, that's a warm, nutrient-rich place. Okay, they're going to sporulate when they're in a place that's not ideal for them. Remember, spore formation is kind of a dormant phase when conditions aren't ideal. They kind of just hunker down. It's a survival mechanism. Okay, so again, with one exception, they're not going to do that in the body. They're going to do that in the environment. Okay, so some examples of Bacillus. Bacillus subtilis is the one that we use in the lab a lot. Again, it's just a soil organism. probably got some on the bottom of your shoe right now. Bacillus cereus or B. cereus, that's known for causing food poisoning. Again, typically not too serious, just make you miserable for a few days. So it's one of really only 2 bacillus that are pathogenic. The other one and the most, excuse me, famous one is Bacillus anthracis. That's a causative agent of anthrax. Okay. So even though anthrax is really rare, in the United States in the years 1976 through 2001, there was only 25 cases of anthrax in those, 25, 26 years. But of course, in 2001, we had the bioterrorism threat, and there were some anthrax spores that were sent in the mail to Congress, and 5 people died. So it's kind of heightened now that, Bacillus anthracis is a, weapon of bioterrorists. So we better kind of be on our toes and understand a little bit about that organism. Okay. There was another case in the late 70s I think it was in a bioweapons facility in Siberia, where at least 100 people were killed when they accidentally released some anthrax spores. And so, again, just an example of the killing power of this disease. All right, so let's look a little bit closer at Bacillus anthracis. Where do you find it? How does it get passed on to other people? Well, it has a soil reservoir. So again, that's because it sporulates. It's going to just kind of hide out in the soil. It's definitely more prevalent in some areas of the world than others. And, like around here, it's pretty much unheard of, but I think like in the Dakotas, they have a little more problem with that. Middle East, a little bit more problem with anthrax. It's considered a zoonotic infection. So humans, we don't pass it to other humans. The way that we get it is by being in close proximity with animals. Okay, so people don't transmit to other people. Okay, so we're not going to have an anthrax pandemic, most likely. All right, so we'll talk about how people can get it when they're in association with animals. Okay. I suppose that it maybe has happened if you try really hard, one person to give it to another person, but it's not going to naturally happen. All right, some virulence factors. Just mention a few here. The organism does make a capsule, but it's a really kind of unique kind of capsule that's mostly made of a protein, of a polypeptide called poly-D-glutamic acid. Remember, most of the time capsules are polysaccharides. They're made of carbohydrates, but this is a little, I think, kind of beefier kind of capsule, a little bit even more protective against phagocytosis from our macrophages, dendritic cells, and helps the organism resist digestion. So those complement fragments like C3b are not going to be able to bind to that outer capsule. Okay, and the capsule, remember, the organism is going to make a capsule when it's inside a host. Okay, not in the environment. All right, so it sporulates in the environment, but when it's in the host, then it will make a capsule. And by the way, if you stain anthrax, it has a very characteristic kind of boxy shaped rod to it. That's, again, kind of, when you see the boxy shape, you think anthrax. Okay. A couple of exotoxins. Actually, the organism makes 3 different exotoxins. One just facilitates the entry into the cell, the other 2. And those toxins do exactly kind of what they're named for, so that's kind of handy. Edema toxin causes swelling of tissue in the tissues. say swelling of tissues. Okay, so really kind of an uncontrolled inflammatory response. And lethal toxin does what you think it would do. It causes cell death. Okay. Another way to say that is that it causes tissue necrosis. So necrosis means, death of cells. Okay, so we'll see some examples of what that does to the host skin here in just a second. Okay, so there's 3 different ways that you can contract anthrax, and it depends on the portal of entry as to how that disease will manifest itself. Okay, so the best kind of anthrax to get, if you're going to get anthrax, is cutaneous anthrax, because that's the kind that's least likely to kill you. All right, and the portal of entry here is through some kind of break in the skin. So I guess we'd probably consider that, through some kind of wound. Again, if the skin is not intact, that is probably a parenteral portal of entry. Okay, so again, the spores are going to enter through some kind of break in the skin. When could that happen? It's going to be when somebody is working very closely with an animal that has anthrax. I was watching a detective show a couple weeks ago, and I got so excited because one of the lead characters had got anthrax from brushing up against a barbed wire fence. And there had been, an animal that in close proximity that had done that, and they got anthrax. And I was like, wow, this is so exciting. So when it enters through a break in the skin, it causes some kind of nodule formation. So a nodule, that just means a kind of an aggregate, abnormal aggregate of cells, or abnormal growth. Okay, so kind of some abnormal growth of our skin cells, and then an eschar forms. All right, so that's a term, it's probably unfamiliar. An eschar is just a black scab. Okay, so you can see the picture here. It looks like a hand model that has anthrax there. I don't know how they found somebody that had that. So anytime you see blackening of the skin, that means that skin is dead. All right, so but usually these eschars, they heal up on their own. They're going to scar, but most of the time they heal up and without any treatment at all. But about 20% of the time, that organism can get in the bloodstream, and it gets systemic. So when that happens, then there's a potential for the patient to die. But it's kind of rare for that to happen. Okay. So again, it's the toxin that has the potential to go systemic. And when that happens, then again, low, about 20% mortality without treatment. Who's going to contract that? People that are working closely with animals. Okay. And it's uncommon in the US because we vaccinate our animals if there's been any kind of anthrax in that animal population. We have a good anthrax vaccine. Okay, the second kind of anthrax, you can contract anthrax if you inhale it. Okay, so we call that inhalation anthrax, sometimes called pulmonary anthrax. This is also known as wool sorter's disease. Okay, so if you've ever, personally, I've never sheared sheep, but I've seen pictures of it being done. And when it's done inside, it looks like a really dusty sort of thing. Okay, so if you can imagine, this wool and lots of things flying around in the air, if the sheep have anthrax or their spores in their Then you can inhale the spores. Forgetting to turn off this notification. Okay, so when, I know we're about out of time here, but when those spores are inhaled, 2 phases of the disease, it starts out very mild. So the prodromal means this is kind of like flu-like symptoms. Okay, nothing to suspect that you have something that potentially will kill you. Sometimes patients feel good for a day after they have those flu-like symptoms, and then bam, the invasive phase hits them. Really high fever, fluid filling up their lungs. They can have such inflammation that they go into shock. That lethal toxin especially acts on macrophages. All right, so that's a problem. When the macrophages are dying, the macrophages aren't able to present that antigen to call in, the adaptive immune system. Without treatment, it's almost always fatal, even if treatment at that part at best 50%. Okay, so we'll finish up anthrax next time. I will get the next set of things posted. I'm still tweaking them a little bit. Hopefully I'll get that posted by tonight or the latest tomorrow. Okay, have a good day.